Hello students, in the present video we will study about the two topics of the plant embryology. One is the embryogenesis in dicotyledons and monocotyledons. Second is the polyembryony. So we start from the embryogenesis in dicot. In a typical dicotyledons, example here the example is of capsula bursa pastoris is given. In a typical dicotyledons, the type of embryo development, the zygote enlarges in size, secretes its own cellulosic cell wall, divides by a transverse division, and that transverse division producing the two unequal cells, the larger basal cell, which is called the suspensor cell, which lies towards the micropylar end of the embryo sac. The smaller terminal cell, which is called embryonal cell and which lies toward the chalazal end of the embryo sac. The basal suspensor cell divides by transverse division, and the terminal embryonal cell divides by longitudinal division to form a tetrad. To form a tetrad, 4-cell stage. Out of the two cells formed from transverse division of the suspensor cell, the lowermost cell towards micropylar end enlarges in size to form a vesicular basal cell. VESICULAR, vesicular basal cell. The other cell divides by a variable number of transverse division to form a long suspensor consisting of a row of 7 to 8 cells. Each of the two terminal cells of the tetrad stage of proembryo divide longitudinally, forming 4-celled quadrant stage. That is 2-celled. After that is the 4-celled quadrant stage. That is this. Each of the 4 cells of quadrant stage then divides by transverse division to form 8-celled octant stage. To form 8-celled octant stage. Each of the 8 cells of octant stage then divides by periclinal division to form an outer protodermal cell to form an outer protodermal cell and an inner cell and an inner cell. After this, the 8-celled octant now becomes 16-celled, 8 external protodermal cell and 8 internal cells. 8 proto 8 external protodermal cells and 8 internal cells. The 8 protodermal the 8 protodermal cells divide by anticlinal division. Here students listen, the 8 protodermal cells divide by anticlinal divisions to form the embryonic surface layer. To form the embryonic surface layer means to form the epidermis. The, a, the 8 inner cells divide by different planes. The 8 inner cells divides by different planes to form the ground meristem and procambium of cotyledons, hypocotyle and plumule कि जो अंदर वाली 8 cells which are divided in different planes and form or produce the ground meristem and procambium of cotyledons, hypocotyle and plumule. Next, the suspensor cell situated at the terminal end of suspensor situated at terminal end of suspensor. Terminal end means in contact with the globular pro-embryo is called hypophysis. That is see here. The hypophysis divides by transferase and longitudinal divisions to form 3 tiers of cells. कौन कौन से टायर है? 3. The cells of inner tier which are towards the embryo form the cortical initials of the radicel means embryonic root. Next, the cells of middle and next outer tier form the initials of root protoderm and root cap. Root protoderm and root cap. So all the cells derived from the hypophysis gives rise the root apical meristem. At this stage, इस stage के ऊपर the developing embryo is globular. And shows the radial symmetry. And it is called proembryo. And this is called proembryo. The proembryo is transformed into embryo with the development of radicel, plumule and cotyledons. Radicel, plumule and cotyledons. There are 2 cotyledons arise from the two ridges of tissues, that is this ridge, and produce the this. The two cotyledons arise from the two ridges of tissues appeared in the distal end of the proembryo, so that the globular embryo converted into or becomes heart-shaped embryo. Heart-shaped embryo. Next, cotyledons grow rapidly, whereas a few cells between the two cotyledons differentiate to form a dome-shaped plumule, means that is the future root apex. Student, the embryogenesis of the dicot is finished with this slide, and the questions may become 3 to 5 marks. So you attempt with the diagrams. Next, embryogenesis in monocote. In monocotes, ये कहते हैं कि the early phases of embryogenesis up to the formation of in means embryogenesis in monocotyledons के केसेस के अंदर कि अर्ली फेसिस जो स्टार्टिंग के फेसिस हैं of embryogenesis up to the formation of globular proembryo are very similar in both dicotyledons and monocotyledons, दोनों में similar है. In monocotyledons, about one half of the terminal cell derivatives function as root apex, whereas the remainder of the cells कि टर्मिनल सेल्स का जो अबाउट रिमेनिंग जो हाफ है, so a rapid rate of cell division in the formation of single terminal cotyledon. So it is the monocotyledon. Students, embryogenesis monocotyledons, there is a great variation कि मोनोकोटिलिडोन्स में कोई भी टिपिकल एग्जाम्पल सेट नहीं हो पाता, क्योंकि देयर इज ए ग्रेट वेरिएशन इन द डेवलपमेंट ऑफ एम्ब्रियो इन द मोनोकोटिलिडोन्स. हाउएवर, द एम्ब्रियो डेवलपमेंट इन Sagittarifolia, that plant is a member of the family Aliceaceae, can be regarded as a generalized example कि उसका ये generalized example दिया गया है. In this, the different stages of the embryo development in the monocotyledons. In this, starting from this first diagram, the zygote enlarges in size, secretes its own cellulosic cell wall, divides by a transverse division, resulting in the formation of two unequal cells. The larger basal cell, that is the suspensor cell you see here, toward the micropylar end, and a small terminal cell, that is the embryonal cell, which are lies toward the chalazal end of the embryo sac. Similar in the dicotyledons or embryogenesis in dicotyledons. The basal cell does not divide further. The basal cell does not divide further. इट इनलार्जेस इन साइज, डिवाइड ना होकर के वो अपना साइज बढ़ाती है, एंड becomes large vesicular suspension filament, sorry, does not divide further and becomes large vesicular suspension, whereas the terminal, जो ये सेल है terminal, whereas the terminal cell means embryonal cell divides by successive transverse divisions, forming or producing three-celled filament. Three-celled filament: a large basal cell, a middle cell, and a terminal cell. And a terminal cell. The middle cell divides by a number of transverse and vertical divisions to form several superimposed tiers of cells. Several superimposed tiers of cells. These cells form the plumule, that is this. These cells form the plumule, that is the stem apex or shoot apex, hypocotyle, radical means root apex, and rest of the suspensor. The terminal cell, that is this, divides and redivides in different planes to give or produce a cotyledon. There is a single cotyledon. In monocotyledonous embryo. In monocotyledonous embryo. Student, that is with this slide, the embryogenesis in monocot is finished. Next, that is the polyembryony. The question on the polyembryony may become from 3 to 5 marks. What is the polyembryony? Polyembryony, that is means occurrence of two or more embryos in one ovule. It was first noticed by Leeuwenhoek in 1719 in the seeds of orange. In a seed with many embryos, usually one embryo matures, जिनके अंदर मैनी, उन सीड्स में जिसमें मैनी एम्ब्रियोज है, मीन्स पोली एम्ब्रियो नहीं है, उनमें वन एम्ब्रियो मैच्योर होता है एंड रेस्ट एम्ब्रियोज डीजनरेट ड्यूरिंग द कोर्स ऑफ डेवलपमेंट. रीजन कुछ भी हो सकते हैं, वो आगे ड्यू टू द रैप मीन्स स्कैरिसिटी ऑफ द न्यूट्रिएंट्स एटसेट्रा. According to Ernest and Skinner, polyembryony may be true or false, which is depending upon the embryos, whether the embryos arise in the same embryo sac or in different embryo sacs in the same ovule. So students, there are various types of the true polyembryony. That is, first, that is the cleavage polyembryony. Do you understand the cleavage? In this, the cleavage of the zygote or earlier stages of its development or proembryo into two or more units. Example है निकोशियाना, rustica, etc. Cleavage polyembryony is common in gymnosperm, but rare in the angiosperm. In Erythronium americanum, first division of the zygote is normal from embryonic mass. Many cells at distal end form the separate many cells of the distal end produce separate embryos. In plants, isotoma and exocarpus, the additional embryos are produced from suspensor cells of the proembryo. Next, that is the embryos from cells of embryo sac other than the egg. In this, the embryo may appear from synergids and antipodal cells in the embryo sac. Synergid और antipodal cells of the embryo sac से embryo का production होना. The synergids may be fertilized by sperms from an additional pollen tube. From an additional pollen tube means किसी दूसरी pollen tube, किसी दूसरे pollen grain के pollen tubes. और develop without such fusion और बिना किसी fusion के. In Argemone mexicana and Phaeolus vulgaris, the additional embryos may appear from unfertilized synergids. Unfertilized synergids. इस प्रकार वो अनफर्टिलाइज्ड synergids से जो एम्ब्रियो का प्रोडक्शन होना, that is haploid in nature. And the embryo produced from the antipodal cells develop less frequently but produce in Almus americana, Allium odorum. ये कहा जाता है और रिपोर्टेड है all the antipodal embryos may not remain viable. May not remain viable. Next, that is the embryos arising from the cells outside the embryo sac. The cells of the nucleus and integuments have also been noticed to develop into embryo. Example citrus है, mangifera है. In Spiranthrus, additional embryos have been reported to be developing from inner cells of inner layer of integument. जो inner cells of inner layer of integument. Such embryos, इस प्रकार का जो embryos है, subsequently come to lie in the embryo sac and are nourished by the endosperm. Next, that is the embryos from endosperm. Embryos which are developing from the endosperm have been reported in Balanophora, Alnus. Javier Ernest ARNST in 1913 found that such embryos develop from egg. कि इस प्रकार के जो एम्ब्रियो है वो एग से डेवलप होते हैं. And got embedded in the cellular endosperm. Next, that is the spontaneous and induced embryony. Spontaneous and induced polyembryony. Spontaneous, as you knows the meaning of the spontaneous and induced. To start from the spontaneous, the polyembryony which are induced naturally or naturally occurring polyembryony. and the induced polyembryony, which are induced experimentally or artificially. The spontaneous polyembryony is of two types. First, that is the gametophytic, second is the sporophytic. In the gametophytic, the polyembryony arising from the gametic cell of embryo sac, एम्ब्रियो सैक की जो गैमेटिक सेल है उससे अराइज होना, प्रोड्यूस होना. इन द स्पोरोफिटिक सेल, द पोली और द एडिशनल एम्ब्रियोज अराइजिंग फ्रॉम जाइगोट, प्रो एम्ब्रियो और इनिशियल स्पोरोफिटिक सेल्स ऑफ द ओव्यूल, जिसमें न्यूसिलस है, इंटेगुमेंट्स हैं, and the embryo development can also be made in culture medium means induced polyembryony. The embryos developed in culture medium are known as adventitious embryos, somatic embryos, supernumerary embryos और एम्ब्रॉइड्स कि जो कल्चर मीडियम में एम्ब्रियो डेवलप किए जाते हैं वो एडवेंटिटियस एम्ब्रियोज, सोमेटिक एम्ब्रियोज, सुपरनमरी एम्ब्रियोज और एम्ब्रॉइड्स कहलाते हैं. द न्यूक्लियर एडवेंटी polyembryony shows the great importance or significance in the human life or in horticulture. The adventive embryos provide क्या सिग्निफिकेंस क्या है कि इट प्रोवाइड यूनिफॉर्म सीडलिंग्स ऑफ पेरेंटल टाइप, यूनिफॉर्म सीडलिंग्स ऑफ पेरेंटल टाइप. द न्यूक्लियर एम्ब्रियोज आर फ्री फ्रॉम डिजीज. In addition to this, the nuclear embryos are free from disease. The nuclear seedling of citrus provide better clones than the cutting. Than the cutting की बजाय जो nuclear embryos है which are better, वो जो better मानी जाती है. ये कहा जाता है कि cuttings from the lateral roots and nuclear seedlings develop better root system, means that is the tap root. So students, the polyembryony is finished with this video, and the questions may become 3, 4 or 5 marks, or maybe 1 marks. In the 1 marks, that is the ask about the terms. Thanks.